My pleasure as Dean of University College to welcome you to this fourth in our series of the MLA Lecture talks, the theme of which this year is rhythm. I might say that for me, and maybe for many of you, coming to these talks in this room at this time every Saturday morning for the past 4 weeks has given my life a kind of rhythmic quality that doesn't quite have in other months. If you accept the words of the Oracle Wikipedia, the term rhythm can refer to any regular recurring motion marked by the regulated succession of strong and weak elements. And this regular recurrence can apply to a wide variety of cyclical natural phenomena, having a frequency of anything from microseconds to many years. We hope that these talks have been some of the strong elements in the rhythm of your experience in this month and this week. Our speaker today is Michael Hughes, who joined the faculty of the Washington University Medical School as assistant professor in the Division of Pulmonary and Critical Care Medicine in the John T. Millikan Department of Medicine in 2017. Professor Hughes holds a BS and an MS in Biological Sciences from Stanford University and a PhD in Biomedical Sciences from the Harvard Medical School. I also note that Michael is one of the organizers of a group that has the interesting name, the Hope Center Clocks and Sleep Club. When I read the name of that, I wondered, what kind of club could that be? First, I imagined late Friday afternoon meetings with tired academics nodding over glasses of sherry as they listened to talks about the invention of the mercury pendulum. And then I imagined, or I hoped, that they might offer tutorials in which my students might actually learn how to get some sleep and make it to my class on time. But in fact, the Clocks and Sleep Club is a serious research group that aims, among other things, to promote work in the role of circadian rhythms in neurodegenerative disease. This is, of course, a very important subject since neurodegenerative disease can take many forms that impact life in significant and serious ways. Today's Michael's topic is in that vein, titled Tempo and Harmonics, Orchestrating Circadian Rhythms in Tissues Throughout the Body. Please welcome Michael Hughes.
Thank you. So thank you very much for that kind introduction, and thanks for the opportunity to speak here today. Regarding Clocks Club, or the Hope Center Clocks and Sleep Club, for those of you who are students at the med school, we actually offer that as a one-unit course. We meet once a month, and we discuss papers, we discuss current research, things like that. If you're interested, I can get you on the mailing list, send me an email. We welcome any and all. The room is maybe 3/4 full right now, so there's plenty of room for other people. So what I'm going to tell you about today is some research that my lab has done on the molecular basis of circadian rhythmicity. And this is going to be an interactive talk. And so my question is, in a show of hands, how many people used an alarm to wake up this morning? All right, let's call that 30%. How many people, this is a Saturday, so that's kind of unusual. How many people use an alarm on a normal weekday? Okay, now we're up to 50%. Fantastic. That hits about what the American average is. So almost all of us, or more than half of us at least, are using an alarm clock every morning, or your phone, or whatever, in order to wake up. Now, I don't use an alarm clock because I have 3 kids. And so I have 3 daughters under the age of 9, so we like Frozen a lot. And I think there's a little bit of truth, there's a little bit of wisdom to be found in this clip from Frozen. Did you catch the trick she did with the eye there? Yes, that has happened to me. Not this morning, but this is how I wake up almost every morning. How many people have had the eye trick? right? Lots of people. It's a thing. So Anna is actually normal. This is how human beings are supposed to be. When the sky's awake, we're supposed to be awake. So what I'm going to tell you about are some consequences of the fact that we live on a spherical globe that rotates. And as a consequence of this rotation, there are daily reproducible cycles of light and dark, temperature, social interactions like Anna waking us up, things like that. So what I'm going to tell you about today are the reasons why Anna is awake when the sky's awake. I'm going to tell you a little bit about how this happens at a molecular level with a particular focus on the 2017 Nobel Prize for Physiology and Medicine. I'm going to tell you a little bit about that and how it relates to the work that we do. And finally, I'm going to tell you about how our work, the work going on in my lab and other labs like mine, influences chronotherapy, which I think is a very interesting idea, which almost everyone should be exposed to. So the first question here is, why is Anna awake when the sky's awake? And it turns out that she's not the only one. So hamsters, this little cute guy here, they love to run on wheels. It's just the best thing ever for them. People have actually shown it's an addictive behavior for rodents, running on wheels. So, for recreational drugs or gambling for humans, the running wheel is exactly the same for hamsters, which is pretty cool when you think about it. Anyway, we can do experiments with that. So what we do is we hook up an instrument to monitor when the hamster is running on the wheel. And I wish I was just a little bit taller. If I was maybe 8 feet tall, I could get to the top here. What you see is in every single row here, we have 2 days of data. And each one of these hash marks is a period of time in which this little hamster is running on his wheel. You can see that this covers weeks and weeks and weeks and weeks of data. Now up at the top there, the yellow boxes, that's when the lights are on in this little rodent's cage. And you can see that for the most part, he's not running at all. That's what rodents are supposed to do. They're the opposite of Anna. When the sky's awake, they're asleep. Now, when the sky is asleep, when it's dark, you can see that this little hamster is running like crazy on his wheel. Okay. So that's a pretty robust observation. That's something which we see in all manner of different rodents. We see it every single time we do the experiment. There's no ambiguity there. It doesn't really tell us what's going on with the animal's internal timekeeping, because maybe the animal is just seeing light and going to sleep. Maybe he's just seeing darkness and waking up. So we do a different experiment. We put this animal in constant darkness. So there's no light whatsoever. For rodents, they actually prefer this. So it's not stressful, it's not harmful to the animal at all. For humans, it's a different story. And what you see there is that this little rodent is still rhythmic. He's still running on his wheel in a very predictable fashion, even though the lights are off. So once the yellow bars have stopped, everything after that is constant darkness. You can see that this rodent is still running in little bursts that have 24-hour periodicity. So that's telling us that it is not simply the presence or absence of light which is making this rodent run. It's some sort of internal timekeeping mechanism. And you can see that because this shifts forward just a little bit every day, we can infer from that that the internal clock of this hamster is slightly faster than 24 hours. It's about 23.7 hours, as a matter of fact. Most rodents are kind of in that neighborhood. Some of them are different. Fruit flies are a little bit faster as well. Humans, interestingly enough, have slightly long clocks. So there's a bunch of people way back in the 50s and the 60s who put themselves down in salt mines or coal mines or things like that and lived there for months at a time, and with constant temperature, constant light, and all that. And what they found was that for humans, our endogenous clock is just slightly longer than 24 hours. Who would subject themselves to this experiment? I don't know. You have to be really passionate about figuring stuff out to do that sort of thing. I mean, they would live down there for 3, 4, 5 months, which doesn't sound like a heck of a lot of fun for me. I'd rather do the experiments on rodents. In any case, humans' clocks are slightly longer than 24 hours, which is one of the reasons why, for many people, it's easier to stay up just a little bit later every night and wake up just a little bit later every morning, unless you have your alarm clock or classes to go to or classes to teach or something like that. It's also the reason why many people find it easier to fly west than to fly east. Right? I see people nodding. I'm not the only one who's found that to be the case. So this simple experiment, this rodent who's addicted to running on the wheel, can tell us a lot of interesting things. It can tell us that we have an internal clock which directs locomotor behavior, and it directs it in a way that does not depend on external cues. It does not depend on the presence or absence of light cycles. So the next question for a neuroscientist, which is what I trained as, is where in the brain is this actually occurring? So this is a slice through a rodent brain. It's kind of through this direction. So you're looking face on at the rodent's brain. The thalamus is right there. You can see these two little nuclei right here. They're called the suprachiasmatic nuclei. That is the locus at which the central circadian oscillator directs everything else that's going on. If you look at a cross-section of Anna here, a cartoon of Anna, a cartoon of a cartoon, you can see that the suprachiasmatic nuclei is right down there at the base of the hypothalamus. It sits right above the optic nerve, which you might recall is relaying information from the eye to the thalamus and then back to the cortex. So the suprachiasmatic nuclei is perfectly situated to detect light signals, and because of its proximity to the pituitary, to the hypothalamus, things like that, it can further direct various physiological behaviors throughout the body. And so I will also just back up very briefly and say that even though there's this internal clock which is driving rhythmic behavior in constant darkness, when there's light, you can see that that clock is being reset every single day, which is important because that's how the animal integrates light signals with its own internal clock. That's also why you feel kind of messed up when you fly east or west by 4 or 5 or 6 time zones, because your internal clock is out of sync with the signals that your SCN is receiving from the environment. More on that later. Okay, I want to talk a little bit about the molecular basis of how the body tells time. And in order to do that, I need to very briefly do a recap of molecular biology. So this is the central dogma of molecular biology. Watson and Crick and many, many others formed this essential hypothesis and developed it and elaborated upon it over the subsequent decades since, I think it would have been about 1954. I could be wrong about that. Anyway, the idea here is that genes are encoded in our DNA. DNA is double-stranded, and we use those genes to generate mRNAs. mRNAs are single-stranded, and they are translated into protein. And for a first approximation, protein is sort of the business end of what's going on in the cells. So DNA is turned into RNA, which is turned into protein. Now, in mammals, in terms of rough numbers, we have about 20,000 different genes in our body. There's all sorts of caveats associated with that number, but for the purposes of today, let's just say there's about 20,000 genes in mammals. Now, in any given tissue, we're only going to be expressing about half of those genes, and the amount of mRNA that we make from any given gene is going to determine, in part, the amount of protein which is created. So that is a means of mechanism by which genes can be regulated as they are converted from RNA into protein. So hold that in the back of your mind, because I'm going to tell you how that relates to a central molecular clock mechanism. Now, it's useful to think about analogies here. So when we think about a very simple clock, we have a nice grandfather clock which is available from Amazon. That's where I got the picture. It's a lovely clock. It's kind of modern, right? Anyway, the way this thing works is you take your finger and you slide it back, and so that is a force which is moving this weight, this pendulum, away from its equilibrium. That's the first thing that we need. We need a movement away from equilibrium. Gravity is going to pull this weight back towards the center, but its momentum is going to carry it all the way back through. So there's going to be a movement away from equilibrium, back to equilibrium, and then away from it again. And so the period of this oscillation is something which is useful. We can use that in order to tell time. Now, a key component besides this oscillation from equilibrium and back again is a delay. We need a certain lag. We need a certain amount of time that elapses in order for this to happen. Furthermore, we need a way to read the time. We need an output of this clock. In this case, there's a whole series of hinges and ratchets and gears that I probably couldn't describe in any great detail, but the consequence of that is that these hands move around a circle, and we know how to interpret whether, interpret which hour of the day it is and which minute of the day it is as a consequence of this. Now you can think about the resetting of the clock as similar to light going in through the retina and being integrated by the suprachiasmatic nuclei to reset the molecular clock. Okay, so that is roughly how a simple pendulum clock works. Now, in molecular terms, how would that work? So this is a hypothesis. This is a very famous hypothesis. And I'm going to tell you why it's famous on the next slide. But basically, it's a perfect analogy for what's going on with that previous clock. So we have a gene, which we call period. I'll explain why later, which is being expressed at an mRNA level. So mRNA for period is being made. It's being translated into protein. And then you get period protein. You can see in the capital letters there. Now, period, what it does, this is kind of cool. This is the neat bit. It goes and it actually represses the expression of its own mRNA. So it's this feedback loop. So period protein represses the expression of period RNA, right? And so when that happens, period RNA is going to eventually get used up, and then the protein is going to decay away because everything in the cell, proteins, RNAs, everything, has a certain half-life, and it's going to go away after a certain amount of time. When that happens, period protein is no longer blocking the transcription of period RNA. This gets expressed again, period protein gets expressed again, it turns itself off. That is the clockwork. It's kind of a neat idea, isn't it, right? At a molecular level, we have basically reproduced everything that's going on here. The key features being a move towards equilibrium and a move away from equilibrium, and a delay, right? The delay is generated by how quickly period RNA can make protein and how quickly that protein can repress the mRNA and how quickly everything gets degraded to reset the entire thing. Now, as you've probably guessed, that clockwork takes about 24 hours. That is how the molecular clock works. Now, I should also mention that the way that we figured this out was actually in fruit flies. I've worked for about half of my career in fruit flies, and every single year that I've worked in fruit flies, people have asked me, why work in fruit flies, right? I mean, they're not mammals. You're not going to help anyone, right? The answer is that they're very simple, and you can move very quick, and your tempo of experiments is really fast, so you can play genetic tricks to work out mechanisms like this. As proof of that, the 2017 Nobel Prize went to Jeff Hall, Michael Rosbash, and Michael Young at Brandeis, and Rockefeller, respectively. You can see that if you get a Nobel Prize, they do these caricatures of you. I don't quite know why, but I guess it's an honor. I don't think any of them are. I don't think any of them are in a position to complain. Anyway, all three of these gentlemen and their labs, they used fruit flies in order to basically work out this entire mechanism and then everything associated with it. And there were all sorts of other labs out there doing the same sort of experiments in mammals and in plants and in fungi and things like that. But the nucleus of this idea was forged by these three gentlemen and a few other people who helped as well We can talk all day long about who got snubbed in the Nobel Prize. But no, this was very exciting for me because this is the first time in my life that I have personally known Nobel laureates before they were Nobel laureates. And I know all the people who were snubbed, and I know about all the relationships and politics and all that, which is kind of interesting, and we can talk more about that if you want. Now, the one thing that I haven't told you about is a way to read the time. For this grandfather clock, the pendulum is moving the hands of the clock, and we have a way of looking at that and inferring that it's time to eat lunch or it's time to go to work or whatever it happens to be. Now, this model only covers the essential timekeeping mechanism. It doesn't talk about the output. The output is what I've been working on for basically my entire adult life. So, before we go there, I want to tell you about the biomedical significance of why we want to work on this. So this is Michael Rosbash here at Brandeis. He's the one in the middle, the one with the kind of scraggly beard. So he is having a glass of champagne. There are cameras pointed at him, and he is very pleased with all the attention. And he's going to tell us about how studies of circadian rhythms are going to impact human health. I'll let Michael speak for himself.
Very much yet, because this mechanism has been difficult to attack with small molecules. But I think the reason for imagining promise on the corner is because the clock, the mechanism that we discovered, governs at least half of all gene expression in the human body. And so at least 50% of all messenger RNAs somewhere in the body undergo circadian oscillation. So that suggests, and that's of course why everything from endocrinology, behavior, metabolism, everything falls under the broad umbrella of circadian rhythms. So that's the reason for imagining being able to tinker and ameliorate many different problems from diabetes and aging. Even cancer is impacted by shift work and good circadian timing. So I think that's coming, but not yet.
All right, coming but not yet here. So the key thing here is that circadian rhythms, or the health of your circadian rhythms, are influencing basically every physiological process that you can name, every behavior that you can name, basically any disease that you can name. Michael mentions diabetes, heart disease is a big one, cancer is a big one, any manner of psychiatric diseases, neurodegenerative diseases, all of these are human pathologies which are influenced by the circadian clock. So what we can try to do is we can try to imagine how we can use our molecular understanding of the circadian clock in order to improve the treatment and management of various diseases. So the question is, how is it that circadian rhythms have such a broad impact on so many different tissues and so many different physiological processes? The key thing here is to understand that the circadian clock is organized in a hierarchical fashion. So it's centered, as I mentioned, in the suprachiasmatic nuclei in the brain. That's this point at which we input light information and we integrate that with the molecular clock to instruct the rest of the body what time of day it is and what sort of metabolic and biological processes we should be participating in. Now, the brain is in turn, through a number of very complicated cues, going to communicate with various different tissues. This chart here shows the endothelium, it shows vascular smooth muscle, it shows the heart. Every single one of these tissues has its own internal circadian clock. So basically everything that you look at, the liver, the heart, the lungs, the kidneys, white fat, brown fat, skeletal muscle, your adrenal glands, your spleen, your immune cells, on and on and on and on. Basically every single tissue in your body has its own internal timekeeping mechanism. These are important for a lot of different reasons. The only exception that I can think of are the male testes, and I don't think anyone has the slightest clue why they don't have a clock. But they don't. If you guys have an answer to that, let me know, and we'll do some experiments. In any case, every tissue has its own molecular clock, and each of these clocks look very similar to the core clock, which resides in the brain. Now, if I can get this animation to work just right. Now, every one of these tissue-specific clocks is going to drive the expression of mRNAs, so messenger RNAs, in a rhythmic fashion. So we have a few examples here. So in the endothelium, mRNA expression of pi1 is going to be rhythmic. So it's going to be higher in mid-evening than in mid-morning. DBP is another famous output gene. GLUT4 is another famous one. Every one of these is going to be tissue-specific. So the amount of mRNA being generated is going to be rhythmic in different tissues. That's going to lead to different amounts of protein, which ultimately regulate different physiological processes, such as your heart rate, such as your systolic blood pressure, such as vasodilation, such as the efficacy of TPA. You can see that every single one of these has its own 24-hour rhythm, which is kind of a neat observation. It's a neat observation, but it also tells us something about pathology, because if we look even closer, we can see that various deleterious pathological events, such as myocardial infarction, ischemia, hemorrhagic strokes, those all have their own rhythms as well. So for heart attacks, if you're going to have a heart attack, most people have it sometime between the early morning and kind of lunchtime. So what time is it? It's coming up on lunchtime. So we're all pretty well good for today. Other events, other pathological events have different timings as well. And so the idea here is that there's this very complex interplay where the clock is governing rhythmic gene expression, and those rhythmic genes are in turn governing different physiological processes which generate their own rhythms. And as a consequence of integrating all of these rhythms across every tissue in your body, we are more susceptible to things like heart attacks at specific times of day. That again should be a clue that we can use these molecular mechanisms to tell us something about how to improve human health. All right, here's where I come in. This is basically where everything was when I became a postdoc at the University of Pennsylvania in John Hoganesh's lab. So we did a very simple experiment. Conceptually, this is about the easiest experiment you'd ever want to think of. We wanted to know which genes are expressed in a rhythmic fashion. Easy, right? Except we want to do it at a global scale. We want to look at all 20,000 genes. We want to know which ones of them are rhythmic in any given tissue. And so what we did was we used rodents again. We used mice, and we collected various tissue samples every hour for 48 hours. We collected the mRNA from it, and then we measured gene expression. So you can see up there that we've done this in a number of different tissues. We've done the pituitary, we've done cortex, we've done cerebellum, we've done hypothalamus, liver, brown fat, white fat. Basically everything that I named earlier, we have tested in mice. We purify total RNA, and then we use two different methods. We use microarrays and we use RNA sequencing to simultaneously measure the expression of every single gene within the mouse. I can talk all day long about that, about the nuts and bolts of that if you want, but for the purposes of today's talk, just take my word for it. We can measure every single gene, how highly it's expressed. At that point, we do some statistical analyses, which again, I can talk all day about, but I'd probably need to use the chalkboard, and I'd rather not today. We apply a false discovery correction to sort of normalize everything and make sure that it's reliable. Okay, so what does the actual data look like? So what I'm showing you here are data from the mouse lung. This is one gene. It's a collagen gene. You can see that its expression level is on the y-axis, and on the x-axis is time. So we started collecting sort of middle of the evening for the mice. We went through 2 consecutive days collecting every hour, and you can see that the expression of this gene has a very sharp peak, kind of right around where the lights turn on for the animal. You can see that the dark bars represent night, the light bars at the top represent daytime. So it has one very sharp peak right around the middle of the day. It troughs in the middle of the evening. It peaks again in the middle of the next day. So this is a pretty unambiguous cycling gene. This is the mRNA level of collagen in the mouse lung. Now, I show you a bunch of papers that we read when we were putting together our own paper on this, which all go to sort of reinforce this point that collagen is really important for remodeling and repairing the lung after various damages. So the fact that collagen is rhythmic with a peak in the middle of the day might tell us something about how the lung is acting in a rhythmic fashion to repair itself in response to fibrotic injury, in response to cigarette smoke, in response to viral infection, things like that. To the best of my knowledge, no one has ever directly tested whether rhythms of collagen actually affect fibrosis and things like that. I happen to be in a pulmonary division at WashU, and I'm hoping that I can get someone to do these experiments to test this. I haven't had luck yet. Okay, so this is what the actual data look like, but recall that we are simultaneously measuring 20,000 different genes. So the question is, how many of those genes are actually rhythmic? We're pretty sure that collagen is, but how about the other 19,999? So in order to answer that question, I want you to bend your minds just a little bit. I want you to think about the peaks of this graph as being yellow, and the troughs as being blue, and the middle points as being black. And then I want you to flip this graph in your head so that you're looking at it face on. So we have a sheet of paper. Now you're looking at it face on. So we've turned a 2-dimensional graph into a 1-dimensional graph. And that's what you see right here, where, again, on the x-axis here are time points. These are 48 hours of data collection. On the y-axis, every single one of these is a different gene. So you can see, like right here, for example, I can't reach any higher than this. Right here, for example, this gene is very, 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 very low. And then at the middle of the day, it starts to get active. Yellow is high expression, remember. And then it goes back to blue, and then back to yellow. So 2 peaks of expression, 2 troughs of expression. You see? So if you add up all the different rows on that column, there's about 1,000 different cycling genes in the mouse liver. The lung, I'm sorry, the lung. Have I been saying liver this whole time? No, okay, I just made that one mistake. Okay, in the mouse lung, there's about 1,000 cycling genes. That's phenomenal. I mean, that's telling us that 5, maybe 10% of the expressed transcriptome in the lung is regulated by the circadian clock. It's kind of a cool result. Now, the lung isn't even the number 1 tissue. It's not even the second most rhythmic tissue. The kidneys are number 2. The liver is number 1, which is why the liver was in my head and why I misspoke there. So the liver has upwards of 4,000 to 5,000 rhythmic genes expressed at an mRNA level. The lung, about 1,500, maybe 1,000. Everything else kind of falls off after that. Now, this is kind of cool. We really like these data. We think it's very important for the field as a whole to try to understand how rhythms and things like collagen, or rhythms in these other 1,000 genes, are impacting physiology and ultimately the susceptibility to disease. So what we did was we built a web page. We call it CircuitDB. You can plug in whatever your favorite tissue is. You can plug in your favorite gene, and we will tell you what it is with the stats down here. So anything you guys are interested in, it's up on the web right now. It's been on there for 10 years, or parts of it have been up there for 10 years. And this has been, I think, really influential for the field as a whole. And this is part of the reason why Michael Rosbash was saying that half of all genes in the body are regulated by the circadian clock to at least some extent, because we have all the data and it's available for everyone to take a look at and you can mine it. Now, that allows us to take sort of this global view of how circadian clocks are regulating gene expression. So I have another graph here. This is a tree graph where we're comparing how similar the rhythmic transcriptomes are of various tissues. If you haven't seen a graph like this before, imagine a tree that you're looking at from the top. It's wintertime, there are no leaves. So this is the trunk here, and we have all these branches kind of radiating out from it. You can see that the closer the branches are to each other, the more similar these tissues are at an mRNA level when we're looking at rhythmic outputs. So the brainstem, the cerebellum, and the hypothalamus, all brain regions, are all nicely clustered together. The heart and skeletal muscle, these are both contractile tissues, right? They're clustering together, albeit a little bit more divergent than what we see in the brain. Similarly, white fat, brown fat, and the aorta, the fatty tissues, are all kind of clustering together. So this was a nice sort of reality check that what we were looking at in our raw data was actually real. I should mention that the reason why the aorta is fatty is because it's actually sheathed in fat. And we do our best to try to trim that off when we do the dissections without getting too graphically gory. But it's never quite perfect. And so the aorta always ends up being kind of similar to the other fatty tissues. The lung, the kidneys, the adrenal glands, and the liver are all kind of divergent from everything else for complicated and interesting reasons. So we're pretty confident that our data is telling us what is actually going on in terms of rhythmic control of RNA in all these different tissues. So the big question is, how many total genes actually cycle in the body? And so what we did here was we simply did a plot where we looked at how many different tissues we had, all the way up to 12, and then we projected if we had profiled every single possible tissue, if we had profiled, your earlobe, for example, how many cycling genes would we get? And it turns out that it's well north of 10,000. So greater than 50% of all genes in the body are rhythmic in at least one tissue. That's what Michael Rosbash was talking about in his Nobel lecture. So that's very exciting. And that's telling us something important, I think. At the same time, I acknowledge that that's a very academic question. It's sort of how many angels can stand on the head of a pin sort of question. And people like me are really into questions like this. But in the grand scheme of things, who really cares? Does it matter whether it's 10,000 or 9,000 or 8,000? What does it really matter? And fortunately, we were able to get some really nice press for this paper that we published. You can see that we got a nice write-up in Science. We got a nice write-up in Forbes, of all places, which was surprising. A write-up in BBC Health, a write-up in the New Yorker. This one's my personal favorite because some staff artist put together this graphic, which just tickled me to no end. We got a write-up in The Scientist and even the mighty UMSL Daily. So I should have mentioned that for a number of years, I taught physiology at UMSL. And so this was a big deal at UMSL at the time. So the question is, if this is a very academic question, how many genes are regulated by the circadian clock? Why is Michael Rosbash talking about it in his Nobel lecture? And why is Forbes and BBC and the New Yorker writing articles about this? What's the big deal, right? So here's the answer. The answer is that this has huge implications for pharmacology. Right, so what I want you to do is I want you to imagine this black circle, the empty white circle with the black rim, as being every single gene in your body. The ones that are rhythmic, the ones that are regulated by the circadian clock, are in red. That's a big circle. It's about 45% just based on the data that we have. We can project that out to more. Now, the ones in, what color is this? Is this green or brown or orange or something? Something like that. Whatever color this is, those are genes that are known to be associated with human disease. And so they overlap to a large extent with rhythmic genes. The ones that are blue are targets of drugs. So they encode proteins that we can take a drug to treat. Now that is unfortunately a much smaller circle than the other two, just because the drug discovery is tough, right? I mean, it's hard work, and not every gene out there presents itself as a target for something which can be treated. However, if you look at the overlap of rhythmic genes, genes associated with disease, and genes that are druggable targets, there's about 900 of them. That's phenomenal, right? That means that for these conditions and for these drugs, there's about 900 of them where the time of day at which you take a certain medication is likely to influence the efficacy of that medication. People have known this for years. So statins for blood pressure, right? The first generation of statins, you were instructed to take them before bed because they have a short half-life and their impact wears off over time. And so if you want to get the maximum efficacy of that pharmaceutical, you need to take it at a certain time of day. So what we've done is we've gone through the top 100 best-selling drugs in the United States and Europe, and we've asked how many of them fit into that unity right there where they are targeting a disease-associated gene, which is also rhythmic within our data set. And you can see that there's a whole bunch of things here. The sales in billions on the top and then millions down here are in this column. This is the trade name. This is what they're used to treat. Now, many of these things, many of these drugs, I should say, are treating things which are really unpleasant, unpleasant sort of conditions that I wouldn't wish on my worst enemy. Things like, Prilosec is on there, Lunesta is on there, Viagra is on there. What's the ADHD drug? Focalin is on there. So these are all, important things that we need to think about in the context of circadian rhythms. But at the same time, there's a lot of life and death things on here as well. So these drugs treat COPD, which is, I think, it's the third or fourth leading cause of death in the United States, even more in the developing world. Up at the top there is Rituxan, also known as rituximab, which is a treatment for, among other things, lymphomas. So this is actually a matter of life and death for some people. When you deliver a drug, if it is targeting something which is rhythmic, when you administer that drug may have serious consequences for how efficacious that treatment actually is. And so that brings me to my second to last slide, where I'm going to answer the question, what is chronotherapy? So chronotherapy is using these sorts of data in order to design better clinical trials. So to give one example, a number of people in industry have become very excited about these data because if you guys know anything about the pipeline through which new drugs are discovered, it costs a heck of a lot of money, millions, billions of dollars, to go through preclinical trials. And if you pass preclinical trials, then you have to move on to safety trials. If that works, you have to move on to efficacy. So phase 1, phase 2, phase 3, preclinical before it all. So pharmaceutical companies are spending billions of dollars developing these drugs in preclinical models. If they pass that, they move on to safety trials. If they pass that, so it works in animals, it looks like it's safe in humans, it moves on to phase 2. If it doesn't work in phase 2, the FDA doesn't approve it, and the drug is lost. All that money is wasted, more or less. So the question is, have we been wasting our money because we simply have not been administering the drugs at the right time of day? So industry is very excited that they might be able to rescue some of these failures simply by designing better clinical trials. For example, to make this concrete, kids with leukemia, this is about the saddest thing ever. Kids with cancer is just awful, awful, awful. WashU's Relay for Life is coming up in another month or so, just as another pitch for that. I hope everyone attends. Anyway, kids with leukemia, about the saddest thing ever. You know what time kids get their chemotherapy when they have leukemia? Any guesses? Anyone? Friday afternoon. You know why? so they can be sick over the weekend so they can go to school on Monday. Is Friday afternoon the right time for treating leukemia with these drugs? I don't know. Probably not, I would guess. But no one's ever done the experiment, right? So we don't know. So hopefully the impact of this work will be designing smarter clinical trials that take into account rhythmic variations in our natural biology in order to really get at the essential pharmacological properties of the drugs that we're treating people with. Now, to go back to the cancer example, this can be useful not only in terms of increasing drug efficacy, but also decreasing side effects. So, if you think about a chemotherapeutic, basically we're trying to poison the tumor faster than we poison the rest of your body. The reason why people come off of chemotherapies is because they're too toxic, you're having too many side effects on the liver, on the kidneys, on other regions as well, on your immune system particularly. So the question is, if there are rhythms in how dramatically a chemotherapeutic agent is affecting your liver or your heart or your immune system, then we can time the administration of that drug in order to maximize the extent to which it poisons the tumor while minimizing the extent to which it is poisoning the rest of your body. And hopefully, if we do that smart enough, we can actually save some lives. And my mentor, John Hoganesh, has followed up on this explicitly. I don't have the data in front of me, but he's shown that for pancreatic islet carcinomas, the cancer that killed Steve Jobs, it's treated with a molecule called streptozocin. Now, streptozocin is actually one of those drugs on the table that I showed you previously. What they've shown is that if they administer streptozocin in the evening versus the day, then the impact on the immune system and the impact on body weight loss is considerably better than at the opposite time of day. So that is a very, very attractive avenue for academics and for clinicians and for pharmaceutical companies to follow up on. And with that, I'd like to thank all the people who have helped me out in this work. So this is my lab. We are in the CSR building over in the med school. That's Matt, my postdoc, Jaja, my postdoc, Aaron, my tech, and Christian Scott, my PhD students. I'd also like to thank all of my collaborators, John Hoganesh in particular, my postdoc advisor, who's now at Cincinnati, some friends at the University of Pennsylvania, and my friend Karen Esser at the University of Florida. I'd also like to thank my funding from the NIH, without which none of this work would ever happen. And with that, I'd just like to say it was a great pleasure being invited to speak here today. Thank you all for your time. Mm-hmm. Yeah, happy to take as many questions as you have.
Do you foresee, we have friends who have had amazing recoveries with immunotherapy now on cancer. Do you see this leading to the individualization, if that's the word,
right,
of pharmacology, where one pill doesn't fit everyone who shows
absolutely, absolutely. The really hard part for this specifically is that every one of us have slightly different rhythms, and it changes as you age. So my daughter, who's basically Anna, is the one peeling my eyelids up, and God, I hope she gets out of this phase soon. As you develop into teenagers, your clock shifts later, and then as you hit kind of middle age, you begin to be an early bird again. I've witnessed this in myself. So everyone's going to be a little bit different. And then 20% of the American workforce are shift workers. So flight attendants, people who work in factories, nurses, police, firemen, things like that. That's going to disrupt everything. So the really hard thing in terms of making this sort of work personalized is being able to tell what a specific human being's clock is actually set at. And there's a number of people working on that in terms of instrumentation. People are using wearables like your Fitbits and stuff like that where they're monitoring temperature. You can monitor melatonin and cortisol levels and saliva and stuff like that. There's a whole bunch of different correlates that people are using. And some of my friends who are still at the University of Pennsylvania are developing bioinformatic pipelines to be able to say, in a clinical situation, this patient's body is set to 8 a.m. even though it's 10 a.m. for the rest of us, et cetera, et cetera, et cetera. So we're a long way away, but people are actively working on it. And yeah, and I think the immunotherapies are a fantastic application for that. Yes, sir.
So two questions. This clockwork includes all of our bacteria?
Good question. People are starting to look at that too. The composition of our bacteria changes over the course of the day. It changes over a lot of different things, but the day as well. And John has had his hand in some of that work. Garrett Fitzgerald at Penn, I think, is sort of the leader in that sort of thing. I don't know a lot about it myself.
And the term chiasmatic, suprachiasmatic, that would mean crossover? Does that have something to do with right brain, left brain?
Yeah, well, so if I back up, there's one on each side. Yeah, so I think that the origin of that word is more because it's sitting right on top of the optic chiasm, where the two optic nerves are passing right at that point, and it just happens to be right there. So it's defined anatomically. I saw a question over here. Yeah. Antibiotics, that is a good question. I don't know the answer to that. I do know that vaccines, the time of day at which you get a vaccine affects its efficacy. And honestly, I can't tell you which vaccine and which virus or even which time of day, but there was a paper by my friend at Hartford who showed that you administer a live viral vaccine. He was doing this in mice, but they've followed up on it in humans. In any case, yes. Antibiotics, I don't know. My personal experience with bacterial infections is that you want to take your antibiotics as soon as possible. I don't know that there's been a time of day yet.
Early on in your talk, I was asking, with this light and dark being important factors in these cycles, have you tested blind people?
Yes, people have done that. Blind people have normal rhythms, most of them anyway. And
How do they sense light?
well, they don't, which is the interesting thing. Some of them don't. Some of them do. And actually, within the retina, the ganglion cells that actually send the optic nerve back, in some blind people, those ganglion cells have degenerated. They're basically dead and kaput. But there's another population of ganglion cells that are sitting in the retina, which are detecting light, which are projecting directly to the suprachiasmatic nuclei, which is cool. Yeah, that's a big result. There's a whole spectrum of different ways that people can be blind. Some of them have normal rhythms. Some of them are rhythmic but sort of drift in and out of phase with the rest of us because they're not being reset by light. Others are just completely messed up.
How much of this is terrestrial rhythms, that is the rhythm of the moon around the Earth, the rhythm of the Earth around its axis, around the sun?
Yeah.
Because I guess, there's been a little bit of opportunity to do some space studies, right?
Right. Light is the number one entrainer that we know of for this 24-hour mechanism. Food is a very big one. So my advice for anyone who's thinking about making jet lag better is to setting your meals on the schedule that you want to be on as early as possible. There are people out there who think that the disruption, the reason why we feel so messed up when we're jet lagged is because we have all of these clocks and they're all being reset at slightly different paces. And so if your liver, I woke up in Europe one morning at like 2 a.m. and I was starving. And I'd been there for 3 days, and it was just the strangest thing. And you can't get back to sleep after that, and it's a big mess. So light is the number one in trainer. Food is the number two. Social interactions can be in training. A lot of other sort of things can be in training. Your other question, though, or the related question, is, are there rhythms other than 24 hours? That's an interesting one as well. So one of the things that we saw when we collected these data, which I didn't have time to talk about today, is that there are actually 12 and 8-hour rhythms of gene expression as well. And there are also longer rhythms as well, not necessarily in gene expression, but in different physiologies. Menstrual rhythms are an obvious one. But at the same time, there's this Scottish professor who, during the Second World War, was stationed on one of these islands off of Scotland. He was way up, and I guess he had a lot of time on his hands, where he was looking for non-existent German submarines or something. I don't know what. But he had a lot of time on his hands, and so he would shave every single morning. And because he was meticulous, he would wash the little bristles and then dry them and then weigh them. And he did this for months at a time. I don't know why. He found a 7-day rhythm in the pace at which his whiskers were growing. What does that mean? I don't know. What are you going to do with that information? But yeah, there you go. So I don't know. Maybe Gillette would have something to say about that.
Yeah, I was just wondering, talking about sort of closest humans and mammals, but fruit flies and other non-mammals, I was sort of taught that these rhythms were not very temperature sensitive, and that's sort of odd for chemical reactions.
Yeah. So this is getting into some. I actually have some slides about this. I'm sorry. What?
Can you repeat the question?
Oh yeah, I'd love to repeat the question. The question is, are these rhythms sensitive to temperature? Is that a fair summary?
Yeah.
Yeah. For the most part, not. And it is very strange. If this was a purely chemical reaction, then we would expect you turn up the temperature, the period length would go faster. You turn down the temperature, it goes slower. The anecdote here is that the first person who showed rhythms in fruit flies was looking for temperature compensation, like you were saying, and he was actually up in Colorado somewhere, and he had these little vials of fruit flies, and he had some of them in his cabin, and he had some of them in a stream, which was, basically melted glacier temperature. Fruit flies don't care that much, and their rhythms were basically exactly the same, which is a little bit unusual. Now, the caveat here is that rhythms can be entrained by temperature pulses. So not the actual period length itself, not the clockwork itself, which is robust against temperature changes, but if you hit it at the same time every day with a pulse of heat, then you will see differences in gene expression, and you will see the phase of the clock change as a consequence. All right. Yes, sir.
Again, two things. I don't know whether it would be called an organ or a system, but the craniosacral sac
Mm-hmm.
that kind of is involved in craniosacral therapy, that John Ledger's findings, that seemed to have a different rhythm. He identified a different rhythm for that.
Physical rhythm.
Okay, I'm not familiar. Can you elaborate a little bit?
The other question is, does the clock affect is it a direct effect on individual cells?
Absolutely.
So the tissue result is an effect.
Yes. And in fact, people have put the protein that fireflies use to light up into cells as a reporter for the circadian clock, and you can image a single cell, and you can see it glow and then dim, and then glow and then dim, and then glow and then dim. If I had wireless here, I'd show you the videos. You can get them on YouTube, easy enough. Yeah, sorry. Yes, ma'am.
Thank you. I was hoping that you would talk a little bit about sleep, and as we age, sleep is getting worse, and how
Right.
circadian clocks help or address that?
Right. I'm not a sleep person myself. I would direct your question more towards Eric Musiak or Paul Shaw, who know a heck of a lot more about sleep than I do. I will say that what I do know is that sleep is amazingly important for cognition and for various healing mechanisms. So Paul Shaw at WashU has actually shown that he can give flies Alzheimer's disease. And depending on how well they sleep, the progression of the disease can be dramatically improved. He can lesion fly brains, and they shouldn't be able to remember anything ever again. And if they're sleeping, they can repair it, and if they're not sleeping, they can't. So sleep is amazingly important. I mean, in terms of practical advice, I would say that basically everyone in the circadian field that I know uses melatonin. I do. Does anyone use melatonin? Okay, a few people, right? It is one of these natural hormones that the pineal gland makes as a consequence of our circadian rhythms. It has all sorts of other kind of curious properties as well. It's a major antioxidant. It has anti-tumor properties. It has a whole bunch of other things as well. In order for melatonin to work, though, a lot of people say that it doesn't work for them. Well, the reason, for my mother-in-law, she swears it does not work for her. The reason it doesn't work for her is because she takes it and then she futzes around with her phone, right? So you're telling your circadian clock two very different things. The melatonin is saying sleep, but the phone, the blue light, is telling your suprachiasmatic nucleus it's time to be awake. So you have to be very disciplined in order for melatonin to actually improve your sleep. What? Oh. The question was, what milligram of melatonin? Which is a good question. And this is actually something I know a little bit about, oddly enough. So anecdotally, my postdoc advisor is a big guy. He's about my height, but he's a little bit wider than me. So generously, 220, maybe a little bit north of that. He takes 250 micrograms per day, which is what I take. So supposedly 250 micrograms is how much the pineal gland makes every day. That's how much I take. And for me, it works pretty well. I'm out. I'm gone. It's not a sleeping pill, right? You're just nudging your circadian system. Now, in contrast, my other friend, Karen Esser, who is on the acknowledgments as well, she's maybe 5'2", maybe 5'3", so generously, 110-ish. She takes 10 milligrams per day. And I've heard some other people say that. So with a handful of people that I've talked to, men seem to be more sensitive to melatonin than women. I don't know why. I can't say that anyone has ever tested that. But people do get lightheaded with it. So I would, start small and work your way up until you see an impact that you're pleased with.
Your work is mostly on the micro level. They say that people are comfortable with iambic pentameter because it's the rhythm of the heart. Does your work have any implications for the macro level, pacemakers?
Not presently. I don't know a heck of a lot about the heart. In a past life, I did some electrophysiology on neurons back in graduate school, but I can't say that I have anything insightful to say about that.
You hardly mentioned lungs. Why are you in the pulmonary?
That's a good question, and a personal side actually. But it's a complicated thing. So I worked at UMSL for a number of years, and things were going great. I had a lot of fun at UMSL. I really like what they're trying to do there, this idea of particularly non-traditional students and students from sort of disadvantaged backgrounds and coming in and being a research university and providing things for that. Unfortunately, UMSL went bankrupt, more or less. Do we have any UMSL people in the room? At least one. Okay, so it was a situation where the university was in more or less dire financial straits, and they had to make a decision. The leadership had to make a decision between students and research. And they basically chose the students, which is probably the right decision. But they making their decision obligated me to make a decision to continue my research career at a place which is committed to research, like WashU, for example. And the academic job market is peculiar. Many of you know about this. The reason I ended up in a pulmonary division is because the chief of pulmonology, Mike Holtzman, has a very keen interest not only in circadian clocks, but also in the sort of genomics experiments that I do. And so that's why it was a natural fit to get us together. So moving forward, I think that if I were giving this talk 5 years from now, we'd be talking a lot more about chronic obstructive pulmonary disorders and asthma and things like that. But the data is just not quite there yet. So.
Couple more questions.
Anyone? Yes, sir.
When we think of things that are controlled by light and dark, I think of the animal world.
Mm-hmm.
Because there are times at nighttime when I used to camp and everything, as it would turn dusk and everything, the night animals would go to sleep in the night, birds and everything would come out.
Right.
But I've noticed that when we saw the eclipse, my wife was out for the eclipse, and it started to take effect, everything started to die down, even the birds, everything.
Right.
They were approaching night.
Right.
And then when the sun came back out, completely. So
right.
whenever we go to, the days get longer and the nights get short, does the animal kingdom, do you think, would they reset their clocks during this period? Otherwise,
absolutely. And that actually gets into this gentleman's question back here, this idea of yearly and seasonal rhythms as well. And so that is one of the big reasons why we have this mechanism where we can re-entrain the clock, right? You have a grandfather clock, and every now and then you need to retune it. You need to make sure that it's set back to the right time. Now, as we move up to higher latitudes, right, the days during the summer are longer and the nights in the winter are longer. Animals need a mechanism by which to reset their molecular clock to do that. Now, here's the cool thing. People have done genetic studies of humans. So they've done epidemiology sorts of things, anthropology sorts of things, actually, where they are looking at mutations within clock genes in different populations of humans. And it's a cool bit. So Northern Europeans, like my ancestors, would have left Africa, left equatorial Africa, 100,000, 200,000 years ago, something like that. And there are mutations within the genes involved in the molecular clockwork which make it easier for people in northern climes to readjust their clock to shorter days or longer days or things like that, which is pretty cool. Right now, it comes at a cost, right? Because, the ancestral Homo sapiens are going to be residing just about on the equator, right? And if you look at African populations that really haven't moved around much in the last 100,000 years, they have different clockwork mechanisms than Northern Europeans or Inuits or Eskimos or people like that. Now, it comes at a cost, right? Because these mutations, which help us adapt to higher climes, are also linked genetically to higher incidence of psychiatric disorders, bipolar, major depression, things like that. So there's no free lunch ever. And it's also why, a lot of people, my grandmother in particular, have a seasonal affective disorder, right? Some people just get the winter blues. Some people are more susceptible to it than others. As of so far, I think I'm more or less safe, even though my grandmother had that. Yes, sir.
I was wondering about the fact that we live indoors so much. I know when I was doing research at WashU, I was in a lab that was internal with no windows.
Mm-hmm.
During about 3 months of the year, I never saw the sun. I would get to work
Yep.
in the dark, and I would leave in the dark, and I'd never
Yep.
go outside. What effect is being indoors and artificial light and so on?
Nothing but bad. Nothing but bad. Get outside as much as you can. So, in my opinion, the best sleep researcher in the world is off in Germany, unfortunately, for everyone in the room, Till Roenberg. And one of the things that he's said is that it's not just that you're not getting enough sleep, but you're also not getting enough sun. So he really encourages, and I'm willing to take his word for it, he really encourages people, particularly as you move further north, to get as much sunlight as you can, UV exposure notwithstanding. Because, your body really does need this resetting mechanism. And so, I encourage it. I encourage you to get outside as much as possible. Fluorescent lights, fluorescent lights, the spectrum of the light is pretty narrow, and it's not doing a heck of a lot for this mechanism. So you need that bright white light.
So on that note, let me suggest that we thank Michael Hughes for trading.
Thank you all.